Hi, this is a presentation on the development of the Gardasil 9 vaccine by Jun Yin, Aditi Dusmili, and myself, Arjun Singh. So human papillomavirus, or HPV for short, is largely responsible for cervical cancer cases in American women. Therefore, the effective virus-like particle or VLP vaccines, Gardasil and Gardasil 9, have been developed. VLPs work by stimulating an immune response but not being able to actually spread infection, and Gardasil 9 specifically is able to protect against 9 types of HPV with a nearly 100% efficacy rate. In terms of the process, first comes discovery, then preclinical and clinical trials, then regulatory approval, and then manufacturing for production, and then finally quality control to see how the vaccine works in the real world.
The manufacturing process consists of fermentation, purification, formulation, and filling. So the fermentation and purification for each HPV type's L1 protein VLPs are done separately, but they are similar, so we are using the synthesis of the HPV type 11 L1 protein VLPs to demonstrate the overall process. So fermentation begins with a single yeast cell with the L1 protein, then seed fermentation, production fermentation, cell harvest with cross-flow microfiltration, and freezing to attain a frozen yeast cell slurry, which will be used in the next step. Purification begins with thawing and cell lysis, which is cell disruption to release the VLPs. Then there's incubation, cross-flow microfiltration in diafiltration mode, and cation exchange chromatography to further purify the VLPs. And for most types, you use disassembly and reassembly of the VLPs with DTT, which breaks disulfide bonds, and that improves stability. And the last step is sterile filtration, which removes microorganisms. So this overall process achieves up to 98% homogeneity of the purified VLPs. And one problem area for purification and subsequent steps in the downstream process is that the VLPs aggregate irreversibly outside of certain pHs and salt concentrations, so those have to be monitored in order to ensure that the VLPs are stable.
So the next step of the overarching manufacturing process of the Gardasil 9 vaccine is formulation utilizing a portable tank, and this step begins with adsorption onto adjuvant and dilution to form the MBAPs. Then a histidine buffer is mixed with alum and saline along with polysorbate 80. The 9 synthesized MBAPs are then added to the mixture is settled and decanted, the type-specific L1 protein concentration are reduced to their targets, and this final formulated bulk that was created is agitated and recirculated. Then, in the filling step, in a barrier-isolated system, the final formulated bulk is filled into vials using an automatic filling machine. Weight is monitored routinely, and upon completion, the vials are removed from the system and checked for abnormalities. They are then placed in trays to store at refrigerator temperature of around 2 to 8 degrees Celsius, and they're ultimately labeled and packaged.
Analytical techniques are used throughout the manufacturing process to determine purity. So STS Page and Western blot are used to find purity of the protein, and other assays are used to determine carbohydrate concentration, amount of residual yeast DNA and RNA, lipid content, and amount of endotoxins.
So equipment used in this manufacturing process can be broken down by categories. So for temperature regulation, dry ice and ethanol baths, room temperature environments, refrigerators, and thermocyclers are used. Next, process tanks, devices with positive displacement pumps, homogenizers, and centrifuges were used for mixing as well as the opposite. Bioreactors, portable tanks, and automatic filling machines in a barrier-isolated system make up reactor design, and peristaltic pumps are used for fluid transport. So while manufacturing is mostly the same on both the large and pilot scale, using more technology and automation on the larger scale, which would not be cost efficient on a smaller scale, saves both time and money while increasing efficiency, evidencing the advantages of automation in large-scale manufacturing efforts. Now, to move on to the clinical trial process, in general, there are 5 standard phases. Phase 0, which determines safety using a small group of people. Phase 1, which determines proper dosages and administration methods using a few dozen individuals. Phase 2, which observes side effects in hundreds. Phase 3, in which thousands are observed to determine efficacy. And Phase 4 for additional research after approval. For the Gardasil 9 vaccine, Phase 2 and Phase 3 trials, which are typically double-blind or open-label randomized procedures, were conducted among 7 different studies in which men and women of all ages were observed to determine immunogenicity and safety.
In terms of licensure, the Merck Corporation had to apply with the Biologics License application with the Food and Drug Administration. This led to, in December 2014, Gardasil 9 being approved for females aged 9 to 26 and males aged 9 to 15. Then, a couple months later, in February 2015, the Advisory Committee on Immunization Practices recommended routine vaccination for children as early as 11 or 12 years old. And finally, in December 2015, after a supplement to the BLA for Gardasil 9, the FDA approved Gardasil 9 for males aged 16 to 26. In terms of distribution, there are really 2 main aspects, the public sector and the private sector. In the public sector, Gardasil 9 is mainly stocked in community health centers and government healthcare, financed by Medicaid, Vaccines for Children Program, the Immunization Grant Program, and the Children's Health Insurance Program. In the private sector, the Affordable Care Act, or ACA, requires ACIP-recommended immunizations such as Gardasil 9 to be covered, which is why most private healthcare plans offer full coverage for Gardasil 9.
Vaccine development is very time-consuming and costly, like Gardasil 9 took more than 20 years to develop. But this can be drastically improved with innovative technologies, for example, using artificial intelligence in the discovery process, which is usually very time-consuming. And you can also apply innovative technologies to make new types of vaccines like current COVID-19 vaccines. And this also applies to technology in the manufacturing process.